Today we will talk about the antiparallel. Two strands of DNA are antiparallel. What it means? What is the antiparallel? We say that one strand is antiparallel to another strand in a DNA molecule. The two strands are antiparallel. What it means? It means that we have two strands. This is, for example, one strand. This is one strand. This is another strand. In this strand, for example, consider this is a phosphate group is present at the carbon fifth of the sugar, that is present on this side of this strand, and hydroxyl group present on the carbon third of the sugar, that is present on this side, whereas the phosphate group is present on this side in this strand, and hydroxyl group present at the carbon third of the sugar is present on this strand. So we can explain like this. It means that two strands, this is one strand of the DNA. Actually, DNA molecule like this. This is, we say that this is 5' end, 3' end, 3' end, this is 5' end. So we can just, for the sake of simplicity, for the sake of explanation, we will take these two strands like this. This is 5' end, this is 3' end. Obviously, this 5' end of this strand, 3' end could be here, and this is 3' end, 5' end here is here. That means the two strands are antiparallel. That is, one strand runs in 5' to 3' direction, other strand runs in 3' to 5' direction. Two strands of DNA are antiparallel. That means one strand runs in 5' to 3' direction, another strand runs in 3' to 5' direction. This is what you mean by the antiparallel nature. Actually, what this antiparallel nature is, we have basically this, we have this 5', we have 5' end. It means that the carbon at fifth position of sugar has phosphate group free. This is called as 5' end. That means we have this one. This is 1, 2, 3, 4, 5. This 5' fifth carbon, this is fifth carbon, and phosphate is attached to this. That means carbon, this is carbon fifth at the fifth position of the sugar. This is sugar. It is having phosphate group free at the end, at the free end. Now what do we mean by 3' end of the DNA? It means that the carbon which is present at the third position of the sugar, carbon at third position of sugar has hydroxyl group free. It means this is carbon first, second, third. This is third. This is hydroxyl group. This is hydroxyl group. This is phosphate group. Hydroxyl group is always present at the carbon third of the sugar, and phosphate group is present at the carbon fifth of the sugar. So these 3' and 5', they represent basically the carbon. This carbon at fifth position, and 3 represents carbon at third position of the sugar. How we can explain this? If we'll take sugar, we'll make a sugar. We have to explain 5' end and 3' end. We'll make a polynucleotide chain. This is one polynucleotide strand. DNA is always made up of two polynucleotide strands. This is one polynucleotide strand. This is another polynucleotide strand. For example, this is one polynucleotide strand. and at the 5' end we have phosphate group, and at the 3' end we have hydroxyl group. This is 1, 2, 3, 4 and 5th carbon. And then this hydroxyl, it is having bonding phosphoester bond with the adjacent deoxy. This is one deoxyribonucleotide. It would have bonding with the next deoxyribonucleotide. That means this is one polynucleotide chain. This is second. This is one nucleotide. This is another nucleotide. These are two joined by means of phosphodiester bond. Then we have phosphate again. This phosphate, this phosphate, phosphate. This is phosphate. This is hydroxyl. So we can say that this phosphate is present at the 5th position. This hydroxyl is present at the 3rd position. So here what are present? Nitrogen bases are present, nitrogen bases are present, nitrogen bases are present, nitrogen bases are present, nitrogen bases are present. So this is sugar, this is sugar, this is sugar, this is sugar, sugar and sugar component. This is one polynucleotide chain. That means it is having sugar component, nitrogen is base and phosphate group. Suppose the sugar component, this nitrogen base is attached to the sugar component. For example, the nitrogen base here is adenine, here thymine, cytosine, guanine, thymine. Suppose these are the nitrogen bases. Obviously, adenine will pair with thymine, thymine will pair with adenine, guanine will pair with cytosine, guanine will pair with cytosine, and adenine pair with thymine. So we have thymine, adenine will pair with thymine. So two hydrogen bonds between the adenine and thymine. There are two hydrogen bonds between adenine and thymine, 3 hydrogen bonds between guanine and cytosine, 3 hydrogen bonds between guanine and cytosine, and 2 hydrogen bonds between adenine and thymine. They represent the hydrogen bond. Now obviously this is a nitrogen base of another polynucleotide. So this nitrogen base is always attached to the sugar. So we have sugar like this. This is sugar. So 1, 2, 3. This is hydroxyl group here, and this is phosphate group here. This is another sugar. That means it would be like this. This is attached to the sugar. This hydroxyl, this is carbon first, carbon second, and carbon third. There is hydroxyl group. This is attached to the phosphate. So it forms sugar phosphate backbone. And this here we have carbon fifth. There is phosphate group. Then in the next deoxyribonucleotide, obviously it would be like this. This is carbon first, second, third. This is carbon first, second, third hydroxyl group, and this attached to the phosphate group. So here is phosphate group at the carbon fifth. This is next deoxyribonucleotide. We have this is one carbon first, carbon second, third. At the carbon third, hydroxyl group is present. This is carbon third, hydroxyl group is present, and this is having linkage, phosphoester linkage with the adjacent deoxyribonucleotide. So it would be having like this. So at the carbon fifth, what is present? Phosphate group is present. So this is carbon fifth. Then we have another nucleotide. This is 1 2 3, this is hydroxyl group at present at the carbon third, and it is this hydroxyl group is attached to the phosphate component of the adjacent deoxyribonucleotide, and this carbon fifth is having always this phosphate group. So see, these are two strands. This is one strand, or we can write here, this is one strand, this is another strand. This is 5 dash, this is 3 dash end, this is 3 dash end, and this is 5 dash end. So we can also represent here, this is one strand, this is 5 dash, 3 dash, and 3 dash, 5 dash. So it means that this, what do you mean by this 5 dash end? It means that the last end volar nucleotide, the terminal sugar, it is having phosphate group free. It is having phosphate group free at the carbon fifth. That means here this is free phosphate group at the carbon fifth. In this strand, in the same strand, we have having hydroxyl group, OH group free at the carbon third. This is carbon third of the sugar. Then in the same next strand, this is one polynucleotide strand, and this is another polynucleotide strand. In the another polynucleotide strand, we see that we have hydroxyl group free. This is hydroxyl group free at carbon third of the sugar at one end, and we have in the same strand we have phosphate group free. At the carbon fifth of the sugar. So we say that this is 5 dash end. We say that this is 3 dash end. It's 3 dash end and 5 dash end. So this can explain the 5 dash end and 3 dash end of the DNA. That means this we called as these two strands are antiparallel. That means they have opposite polarity. That is having opposite polarity. Two strands are having opposite polarity, and this means that one strand is in the 5 dash direction, another strand 5 dash to 3 dash direction, 5 dash to 3 dash direction, another strand is in the 3 dash to 5 dash direction. And this configuration, this topography of the DNA is called as, we say that the two strands are antiparallel. That is, they having opposite polarity. One strand runs in the 5 dash to 3 dash direction, another strand runs in 3 dash to 5 dash direction. So remember one thing you have to remember, that carbon, this phosphate group is present at the carbon fifth, and hydroxyl group is present at the carbon third of the sugar. In the last nucleotide, phosphate group is free as it is present on the carbon fifth, so we say 5 dash end, and on the same end, on the neck, on another end of the same strand, this is another end of the same strand, we have hydroxyl group free present at the carbon third of the sugar. So this is what is called as what do you mean by anti-parallel nature of the two strands of the DNA. Thanks.